everyone will be muted upon entry, and you will need to unmute yourself to ask questions. It may be easier with this large of a group to just submit your questions to the chat. And so Shaunak is on monitoring the chat and will be jumping in if it's a question about clarification, or maybe saving some of those questions towards the end. This webinar series started as a replacement for our surge or our summer rat genomics workshop, but I think this is working out really well. We're getting a lot more attendance and we'll hit a lot more people. So what we do is present it within an hour, and then we will post the video recording of this talk later onto our website. And so I will send out an email within a couple of days. It takes us a little while to just make sure that the recording has what we want on it, and make sure that it is showing us the right amount of data and cutting it off soon enough. And afterwards, I'll send out a link with all that. I'll also send out a link this time with a survey, if you can just rate us on stars, and if you have any suggestions on things to do different, or also if you have any suggestions on topics that we can tackle over the next few months. Rob's presentation is available through the GitHub site. So if you did not get a link to that, I sent out to all those that had registered within the half hour before the talk. So you just need to send me a private chat and I will get that sent out to you. Other than that, I think that takes care of the housekeeping things. I'll go ahead and hand it over to Rob.
All right, thanks very much, Laura. Thanks everybody for joining us.
I make a quick organizational note, Rob. Questions in the chat that we are unable to answer for any reason, are you planning to answer them later in writing, or do we all do it verbally?
I'm happy to answer in writing. Yep.
Okay, so we will post those answers as well. So if your question is not answered right now during this webinar, you will get a response from Rob later.
Yep. We have about 30 slides here that I think are sort of core slides, and then I have another 10 that are bonus slides, essentially. So you can gauge how we're doing for time by how far we've gotten there. Okay, so just a title and a thank you to everybody who's been contributing to this effort for quite a few years now. The webinar series is sponsored by an NIDA P30 grant to Laura and myself and Shaunak and others. And GeneNetwork is also supported by an R01 to Shaunak and myself from NIGMS, and that support has been of immeasurable value, obviously. We also get quite a bit of support from the University of Tennessee. My email is down there. If there's one thing I do respond to pretty quickly, it's a query about GeneNetwork's use, a bug report. Any kind of feedback would be greatly appreciated. Okay, so I think Laura already mentioned our surge workshop, and this is sort of, in some ways, as she said, an upgrade for that small workshop that we had in Colorado last summer. The goals today are basically to take you on a review of some of the data sets we have in GeneNetwork, just to show you how to find data, because it turns out that can be quite tricky. Then discuss quickly how to review and edit the data. The data as published, the data as generated are often not quite suitable for quantitative genetics or any kind of statistical analysis. So you have to actually do a little bit of exploratory data analysis and cleanup. We try to do some of that for you in GeneNetwork before we enter data, but some of the data sets are just so large and complex, and the authors and owners of the data often want their data in the original format. So we have added some transformation and blocking, censoring, and Windsorizing tools that I'll briefly describe. Then onto mapping So Shaunak did a great job 2 weeks ago of introducing you to the arguments regarding causality of mapping. It's a big misfortune, in my opinion, that we call it association studies, because they are not mere association studies. They are actually attempts, not always successful, but attempts to get at causal variance, causal loci that modulate trait variability. And I think Shaunak did a great job covering that. So those of you who didn't see his presentation 2 weeks ago, definitely well worth going back and looking at his presentation. There's a link to it right, I hope you can see right there. Let me switch over to a full screen mode here. So you can see his presentation from 2 weeks ago using Leah Solberg's data from, actually the data were generated back in 2004, I believe, but also in this 2009 paper. And the idea of the whole series is to take you from Qtl analysis. This is a little bit of chutzpah all the way to therapeutics and mechanisms. Today, our intent is really to get you to genomic regions and an initial stab at ranking and evaluating candidate genes within a Qtl. So this is something that's been going on now for close to 30 years. It's still not an art, or it still is an art, I should say, rather than a science. And I'm not in any way going to be comprehensive in the candidate gene evaluation or ranking. And one thing I will not be talking about, but we can add this later, is how we actually evaluate DNA variants in regions. We're getting down to the gene level in today's talk. Okay, so the first thing, I think you can see everything that really matters on the screen, even though I'm recording. Let me make this a little smaller and pull it down here where it won't be quite so in the way. I think that should be good enough for most of the slides. And again, you can just download the slides. The intent of this slide deck, by the way, is so you can follow along. So not only is it a presentation, but it's pretty much a primer in how to use GeneNetwork. Quick introduction to GeneNetwork. So it's been kicking in one way or another since about 1993. I remember Joe Nadeau and I had a conversation, I think in 1993, about some efforts that he was spearheading at JAX. It became MGI. And at the same time, I was noodling around with large Excel spreadsheets with mouse gene variants and mutations. And in 1994, we came out with our basically web service. It was actually a Gopher service. And it has moved forward for the last 20, 25, 26 years in various forms, first known as Neurogenetic Toolbox, because of my interest in neuroscience, then as WebQTL, and now as GeneNetwork. So it's a pretty old beast, but we've done, I think, a good job over the last 5 years with Piotr Prince's and Shaunak's help in upgrading it. So it's now a good software stack. Everything you're going to see today, the actual code is open source. Anybody's free to fork it. Just go to GitHub, look for it, and you can do your own version of GeneNetwork if you wanted to. Most of the data is also open source, not all of it, but I will be showing you some data right now that's unpublished. I passed it by the owners. So this is data from the NIDA P50 rat GWAS group led by Abe Palmer. I have used data mainly from Hao Chen today. And so it's unpublished, but fun to deal with. So finding data, there are basically two main ways to find data in GeneNetwork. You can either do a global search, which is shown at the top of this page. You can search for all phenotypes in any data set that match the term nicotine. If you do that, I think you get about 100 some hits. I put in a whole series of terms there just to make it more interesting for those of you that are interested in cocaine or morphine or opiates or alcohol or cannabinoids or methamphetamines. I would recommend doing one search at a time there rather than concatenating many search terms, because there's a lot in GeneNetwork and it'll find quite a bit. For genes and molecules, this would be typically mRNA assays, but also proteomic assays from Rudy Ebersol's group and a few other groups. There's metabolomic data for certain populations. There's metagenomic data, and there is a very little bit of epigenomic data that's coming in. But when it says genes and molecules, it pretty much means transcripts and mainly arrays, because there isn't all that much RNA-seq in GeneNetwork yet, although we are putting in about 250 RNA-seq data sets for mouse forebrain real soon now. So that's the global way to do the search. That search of all genes or transcripts that match TTC27 will actually come up with nearly 1,500 hits in humans, in mouse, and rat, whatever species. So that's a good way to do the search if you just want to do an omnibus search initially and not get too lost in the weeds. However, getting lost in the weeds is fun. And I'm going to show you how to get lost in the weeds. So you'll see on the left side, it says finding data. Now you have the classic hierarchical menu where you pick a species, you pick a group, you pick a type of data set you're interested in, then you pick a specific data set and use a search term. So if you look over on the right side, you'll see that I've selected human, the HG19 assembly. I've gone to the GTEx version 8 data, which is all in GeneNetwork. I've selected nucleus accumbens, and I've typed in a few genes that are of interest for this presentation. Below, there's some gene symbols that are just annoying, like TH for tyrosine hydroxylase. GeneNetwork has a hard time searching for two-letter symbols, so in that case, you really need to use the combined search field, just to kind of a detail. This community, TH is a big gene, and it is there. It's just a little bit harder to find than three-letter symbols. Let's see, how do I? Okay. Rat data. So we have a lot of human data, an enormous amount comparatively of mouse data, and we're getting more and more rat data. So here's the data set that we will be playing with, working with the rat on the RN6 assembly, which is still current. We're going to be looking at the NIH heterogeneous stock data primarily, and you can see the types of data. We have phenotypes, genotypes, and then mRNA assays for a few regions of brain. There is another population that we've talked about before, the hybrid rat diversity panel and the HXB-BXH rats. That's another group that you can select. And then the group that we dealt with 2 weeks ago is also under group, and that is the data from Solberg and Rudai that I will reprise quickly. But in this case, you can now say, I want to find phenotypes associated with nicotine in the NIH heterogeneous stock animals, and that's how you do that. The mouse data, so this is nucleus accumbens data from Mike Miles's group at VCU, just showing that in this case, we can get an enormous amount of data for one gene in nucleus accumbens for 3 species quite quickly. And it can all go into a collection, as I'll show you. So this is the review section. And let me just escape from this so you can see the whole screen a little bit more easily. I think that's got plenty of resolution. So this is from the webinar 2 weeks ago. This is Leah Solberg data. And I just wanted to show you that if you want to find that, you would go over to select and search and just make sure that you've got the Northwestern University cross between Kyoto Webster and F344. It's a behavioral data set primarily, although as we learned last 2 weeks ago, not entirely. Select the type of phenotype, and then if you wanted to study the same trait that Shaunak took us through, that's its official accession number, NWU_10021. The glucose level after stress is just something I put in there. You don't really need it. If you enter that search, you will then get some basic statistics, and I just highlight, that's in the middle panel here, data for 480 rats with some basic univariate statistics that is well worth looking at because you never know what how messed up your distribution is. In this case, the distribution is relatively reasonable. It really does not need to be transformed. It wouldn't hurt to transform it. It's got a little bit of excess skew, but not a big deal. So if you wanted to go back and work in parallel with Shaughnock's JupyterLab notebook, you could do that. The one difference I should point out is that we've done essentially a liftover of all of the SNPs and markers onto RN6. The original data, I think that was in Gary's QTL collection was still on RN3 or 4. So there will be some differences, but let me just show you that those are not a big deal. So let's just go to the next slide and let me zoom in on this again.
Rob, there's a question from Joe Nadeau.
Sure.
What is the source of the data? In general, contributed from the community, grabbed from other data repositories? Are study conditions included, like husbandry conditions, diet, study design? Could you comment on that, please?
Yeah, sure. Great question. And the honest answer is it's inadequate in many ways. The data, the focus of GeneNetwork, so you see the history of GeneNetwork and the quality of annotation and metadata. The original data sets tended to be exclusively from mouse, and I would say 90% of them from the BXD population in specific. And there are some data sets that are published are reasonably well described either in the publication or in the metadata files available in GeneNetwork. But there are a distressing number of great data sets that never got published that have marginal metadata anywhere. And so I would say we're, if one objectively evaluated the quality of metadata, it would probably fall somewhere between awful and woeful. But there are a few bright spots, and I can show you those. What we are doing, Joe, to rectify this is we're trying to throw responsibility for this important task to the mouse phenome group, for mouse anyway, and maybe even for rat. So Alyssa Chesler and team have a much better annotation pipeline in place than we do right now. We've been focused on the computation and getting the QTLs correct with some neglect of the metadata. So that's where we are. So here is, again, the Solberg data, just as seen in GeneNetwork. And I just want to highlight, I hope you can see my cursor. If not, my cursor is hovering around the word RQTL, or the button, the blue button, RQTL. And this is how you configure the mapping once you have the trait accessible in GeneNetwork. This is a pull down that I'll show you in a minute, but we're asking it to map all chromosomes with 200 permutations. If a stratified permutation was appropriate, it would do so. We're not controlling for any other markers, and we're not controlling for any covariates such as sex, age, etc., which one can do. But if we just keep it simple, and this is sort of the default parameters, and just hit the compute button down here, in about 10 seconds, you will get a map of the sort that you see right here with a, I guess, a significant QTL. This is a permutation threshold based upon 200 permutations on distal chromosome 1. But the topic today is really how to get from a QTL like this to a gene, and this would not be your dream come true QTL, because if you look up here in the blow up, you'll see that our last marker is at about 248 megabases on chromosome 1 in the rat back then, and we have at least 30 megabases of uncharted DNA, this more distal. And all of these little blue lines here are all of the genes in this interval. So it's not one, at some point you just have to say, it's probably not worth trying to clone at this stage. Ideally, you want to be within, if you're lucky, a megabase. More typical would be within about five megabases, and depending on the cross type. If you're using a reduced complexity cross, you can clone a locus from, or identify the probable gene from a distance of more than 20 megabases. But in a typical F2 cross or heterogeneous stock, you really want significantly better precision than this to proceed to the next steps. So let's proceed to the next steps. Okay, so today's focus, and we are at 12:22, so is HS-RAT. I'm not going to go through this in any detail. You can look at this slide. It's an 8-way cross, advanced intercross, heterogeneous stock, started way back when in 82. It's now at generation 87 or 88. It's still being generated mainly at Wake Forest by Leah Solberg and team. And the animals are then being distributed around, I believe, the whole world, mainly here in the US. It's a model, it's a rat model of a human GWAS. There's some exceptions. In some ways, it's tidier than a human GWAS, obviously, because we can control the environment. Anyway, that's just to show you what we're going to deal with. Here's the link to GeneNetwork, to the P50 data, exclusively P50 data, exclusively related to addiction and behavioral traits, impulsivity. A really good team. And if you wanted to check out the website, I think it's just ratgwas.org. So you are here. So we're just starting here, and the idea is to wend our way through this honeycomb and see whether we can results. The first thing to do is to see what kind of phenotypes have been accumulated in GeneNetwork. So I'm just going to use a wildcard asterisk symbol here to just see what's what, and just hit the search button. And what you should get is a total of 508 phenotypes that have been measured. Now, those aren't all independent phenotypes. They'll come in families from one particular group or another group. So it has a complex substructure, but let's just keep it simple. So we essentially have 508 vectors of numbers from up to about 5,000 rats. And of course, those vectors won't have the same cases or controls or just individuals. Some some RNA-seq data may have data for only 77 cases. Other data sets, for example, weanling body weight, will be pretty comprehensive, and there'll be data for more than 4,000 individuals. But you can see how these are structured. All of the HS phenotypes start with the letters HS, and then R for clever acronym for rat, underscore, and then five digits. Those are fixed accession IDs, so you can use those in papers. And if you want to, if you use GeneNetwork, this is a great way to sort of acknowledge us. You don't have to acknowledge the effort in the acknowledgement, but if you just embed some of the links that have been useful to you, that'd be great. Which brings me to the point that it's better to get data into GeneNetwork early rather than late. A lot of people hold off until they have a publication, and that's exactly the wrong time to be submitting data to GeneNetwork, because at that point you're demotivated, you're on to your next project, and we're demotivated because we won't end up with any kind of citation in the literature, or it'll be a lot harder to get it. But anyway, HS data in here, a total of 508. You can then pick a subset of these vectors of data, these phenotypes, and put them into your, essentially a shopping cart, a collection. And you'll see that in the upper right-hand corner, where I've added some cofactors, for example, the sex of the animals, the phenotyping batch, which within each of the 3 phenotyping centers, and then a goofy cofactor, which we find useful for quality control, which is the coat color. If you can't map a locus for coat color, you're doing something wrong. So we put it in there just to make sure that mapping is working correctly and things didn't get all out of order at some point. Okay, so I think in the interest of time, I'll speed through this. Here is a collection with about 17 traits in it, cofactors at the top. And there's one particular trait that I'm going to pick on today, which is this trait. And I hope you can see that, but it says motivation to obtain nicotine, AKA subjective desire for, subjective value of nicotine. My idiot's guide version of this, the way I think about it, is avidity of seeking nicotine. This is data that Hao Chen generated for, over 1,000 animals. We'll see that next. The trait ID is this HSR_10505. And what you're looking at over on the left side is the actual trait description with the phenotype description. And coming back to Joe's comments about metadata, this is where I do attend. I try to get a good phenotype description. It's not in any kind of formal ontology, but it is something that I think most users, including high school students, if they're persistent enough, can parse and figure out what the heck was measured. So I tend to be quite, those of you who dealt with me, I tend to be quite dogmatic about the structure of these phenotype descriptions. Nobody has taken the lead other than GeneNetwork in getting phenotype descriptions in some kind of short form that's actually comprehensible. Underneath the trait description of the phenotype, there are fields here: basic statistics, transform and filter data, calculate correlations, etc. And you can actually see the trait data over here. Here's the page bottom on the right side. In this case, you can see the cofactors: the sex, the color, the batch, the date of birth, the site that the animal spent most of its life at, the date that the work was done, the phenotyping, and the date of death. All of those cofactors can be used in the statistical analysis. And then the value. So the value in this case is whatever Hao Chen and Apurva submitted to GeneNetwork. It is the ratio of, in a progressive ratio test, how gung-ho the rat is at getting a dose of nicotine. And I would let Hao describe that in more detail, but we'll treat it as, it's actually an interesting, difficult, and problematic trait, which is one reason I chose it. In this case, in the next slide, I've just sorted the same data, but now from high to low value. So you can see that in this data set, this value is about 707 ratio. In other words, the frequency with which the rat taps on the active lever as opposed to the boring lever. This scale, though, you can see that it quickly is reduced to 5 here. So we want to know what that actual distribution looks like. Is it anywhere close to normal? So we need to have a look at the basic statistics for this trait. The other thing I've done here in the up in the middle is I've just for fun entered the term albino. So you'll notice that all of these animals happen to be albino, but if I took that phrase out there, obviously there'd be other coat colors. And you'll notice also the export functions and a reset button. I'm going to censor this slide itself. It's a little bit too complicated, and just go on to the next one, which is to show you the distribution of the trait that we just talked about. So this is the seeking behavior, and this is the basic statistics over here, data for 1,248 animals. It's got tremendous skew and kurtosis, and we can't really map it in this form. So it's just way too out of normal to be handled. So here's the histogram. Here's the violin plot, which does not look like a violin. It doesn't look like a banjo, and I don't know what it looks like. And over here is the QQ plot showing that we only really have a small number of animals that are essentially high outliers. So this is something we did in response to a request from Abe. we made sure we could transform these data without messing with the original data in the database. So in this case, I've taken the original vector of data and I've applied a quantile normalization to the data. So what you see over here on the right side is as normal as it can be made given the hard limit at 0. It's clearly not quite normal, but this is actually reasonable for mapping purposes. So we're going to go with that. It's obviously not ideal, but it's as good as I know how to do with the data as given. You can see the transforms there, log 2, log 10, z-score, quantile square root, and you can just flip the polarity if you wanted to. The log 2 and log 10 do automatically add a value of 1 so that they don't generate negative infinite values. So a lot of error checking that we have to do. Okay, so a little jump there, but here we are at 12:33 Central Time, and here's a Qtl map, in this case, for that trait in the HS. It shows you on the left side the actual settings that were used. So we're using a linear mixed model code written by Xiang Zhou at University of Michigan. I actually wrote it when he was in Matt Stevens' lab at the University of Chicago. We haven't done anything too fancy, so we're mapping the entire genome except for X and Y and the mitochondria. We're not we're using the default genotype file. We set the minor allele frequency to 0.05, and we're using the leave one chromosome out method in order not to absorb variance right at the test locus. And then we just hit the compute. And this is an area that we're working on. It's currently painfully slow to do this. So this, I think, took 45 minutes a couple nights ago. Once it's done, though, if you don't change anything, the subsequent mapping is reasonably quick. So you don't, but we'll have this fixed in the next few months. Working hard on it. For those of you who don't do a lot of mapping, so every speckle here, this is sort of in GWAS form. Every speckle is a SNP or variant that's been typed. The y-axis is the minus log p of linkage. So it's a little bit different than the likelihood ratio statistic or the LOD score that Shaunak described in his talk 2 weeks ago. There are many, many papers that I should have read years ago. At Shaunak's insistence, I finally broke down and read them recently, so I actually know what the difference is. And I'll put a link to the Lander and Krugliak and another paper that I think are sort of a touchstone papers that explain these methods. You can see we have something interesting on chromosome 6. So the x-axis is chromosomes with a megabase scale, and we have a peak linkage statistic of about it's actually 5.19 on proximal chromosome 6. We haven't done anything fancy in this mapping. We haven't included any cofactors, and that's a possible criticism of this particular map, and we'll come back to that and see if we can do better or worse if we add cofactors. But that gives you an idea of where we would like to be. You'll notice, unlike your expectation, there is no magic horizontal line here to tell you what's significant and what's suggestive. That takes a permutation, and ideally a stratified permutation. You don't want to permute males with females, or one site with another site, or one batch with another site. And we're working on getting that working in using GEMMA. It does work pretty well with ArcQTL. So stratified permutation can be done in the other mapping methods. There are 3 mapping methods that you can apply here, and it does work pretty well for ArcQTL with stratified permutation. So you can get a feel for where the threshold should be, but not using GEMMA. So here's what we need to do if we want to map with covariates or cofactors. I did this using GEMMA, and you can see that where the red arrow is over on the left side, there's a select covariates button. If you click on that, it will then give you a list of all of the potential covariates, which is anything that you've put in your shopping cart or your collection. So in one of those first slides, I showed you that I put a bunch of cofactors and I think a total of 17 traits into my collection. So I've picked the 2 cofactors, covariates, sex, and batch number. Then I have simply hit that compute button, and we're using GEMMA across 1,248 cases for which we have genotypes from Abe and team. Over on the right side is the same kind of screen, but now for our QTL. So some of the items there are a little bit different. It's got stratified permutation. I've added another covariate there, center, which is irrelevant for this particular data set since it was only generated at 1 center. And here is the same map now with cofactors. So we're a little bit relieved. We see that the locus on chromosome 6 is still there. It's actually a little bit better than it was. So it went from 5.2 to 5.6. That's not a trivial jump. That's on a log 10 scale. That's a reasonable boost by using the cofactor. You can see beneath this Manhattan plot, there will be a long table with the mapping statistics for every one of those markers. And you can see that right now they have goofy names. We don't have the official RS names for these markers, but in the lower right-hand corner, you can see that there's a marker named CHR6:21, et cetera, which is the marker at the peak with a lot of 5.59. So it is now 12:40, and I'd like to move on to actually getting to evaluation of candidate genes. So we've got a region, but we need to zoom in. And how do we zoom in? The way you zoom in is you just click on the number 6. So if you want to look at all of chromosome 6 only, you would just click on that number 6. And then if you wanted to progressively zoom, you'd do what happens in the next slide. So this is all of chromosome 6 now. Notes, yeah, that is all of chromosome 6. Sorry about the color scheme. We'll get blue on gray is probably not the smartest color scheme. We'll get that straightened out in the next couple of weeks so that you can see the SNP test points a little more clearly. At the top, you'll see four colored bands running horizontally. These are zoom ins to different resources. So one of them goes to Laura's Phenogen website. I think the bottom one is to Phenogen. The top one is just a local zoom. And then there's a zoom to the UCSC genome browser for rat and to the Ensemble genome browser. So if you just want to switch over from one browser to another, you could do that. We actually would like to zoom into that interval right around 20 megabases. And here's the progressive zoom of that region. Upper left, you can see a region of about 9 megabases. And the genes now begin to be resolved. They're organized in this funny way, just so that we can cram them all in there. Obviously, they're not arranged that way. They should be linear, but we have to shift them up vertically a bit. You can now see the individual test points. I think there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, about 12 SNPs in that particular interval that's highlighted in pink that I consider a rough critical interval. It's a relatively small critical interval. It's just under a megabase. You can argue about this, but I regard this as absolutely wonderful resolution and arguably the asymptotic maximum sensible resolution. You could say, well, you can never have enough resolution, and that's true, but it doesn't help you. At a certain point, if you know exactly what SNP is causing a phenotypic variance, you still might have to look at 3 or 4 neighboring genes extending over 500 kb. So I'd say 500 kb to 1 megabase kb is sort of the optimum asymptotic resolution that you should try to achieve in QTL mapping. At that point, my sort of motto is, if you need more than 1 megabase resolution, you need a bioinformaticist as a colleague, because at that point you should be able to go through some of the candidate genes. So in the lower middle, I've blown it up a little bit more, and I've highlighted 4 candidates that are, only 3 of them are formally in that QTL or this locus. That RAS, I'm sorry, there's a typo there. That should be RAS GRP3, not GPR3. That is nominally outside of the critical interval, but you could imagine a variant in a promoter or enhancer that's 250 kb away that's actually modulating the expression level of a gene that's outside of the formal critical interval. So take the critical interval and extend it a bit when you're at this scale. So who's in there? So if you just scroll down basically this window, so the window that gives you the zoomed version of the map will also give you what's called the Interval Analyst. And that will show you, in this case, all 17 protein coding and junk genes that are in this interval from 21 to 22.4 megabases on chromosome 6. It's got all of the rat stuff on the left side, then the homologous synthetic mouse regions, and then the homologous synthetic human regions. So this corresponds to chromosome 17 at roughly 75 megabases mouse, chromosome 2 at about 33 megabases in human. And of course, this will depend upon the particular assembly, and you notice the headings there tell you which assembly, HG19 or MM9. Actually, that's a typo. That should be MM10, something I'll have to attend to. We did the liftover about 10 years ago. Shame on me. So we've got some interesting candidate genes here. And now, with 15 minutes left, we need to evaluate these. This is going to be quite cursory. Just let me start by saying that candidate gene analysis is always a messy ad hoc business. And let me just say why. So far, all of our analysis has been minimally biased. There might have been some bias. For all you know, I played with the transform 10 different ways and picked the one transform that just happened to work. So there can be bias, or at least a multiple testing issue, even at an early stage. But I promise you I didn't. I just went for it. This is the first trait I mapped 2 days ago, and what you see is my first pass of it. But now we're going to add all kinds of useful prior data, but this adds a tremendous amount of potential bias, and that's because the literature is tremendously biased. If you just went by the literature, just about every gene that you map would be cancer-related, frankly, just because there's so much cancer data on genes. So there's an inevitable intrusion of bias. That's bad enough, but what's really bad is the pressure of narrative potential. If you map a gene for pain sensitivity and it maps anywhere within 5 megabases of the mu opiate receptor, I guarantee you that you're going to be making a story about the mu opiate receptor, even if it's not the right gene. So you just have to buck this internally, and it's extremely difficult to buck it internally because your success as a scientist is judged by not the data quality, but the number of snappy papers you have in Nature Genetics, Cell, Neuron, etc. So I can't emphasize this enough. There's no right and wrong, but just beware of narrative potential, especially if you intend to clone something or try to tinker with its genome or the sequence, because there's a high risk of having a wonderful story that's not true. And it may be a wonderful story, it may get you into Neuron or Cell, but so from that point of view, it might be a good thing for your career and get you a grant, but keep it in mind. So in this case, in the lower left, I've put in the names, the symbols of the genes that were in that critical interval. And I'm going to look at their expression in the nucleus accumbens, which is a part of the rat brain, mouse brain, human brain that's involved in seeking, addiction, affect, many things, but it's probably the single best known region for brain region for its association with addictive behaviors. So we're going to look at that. So here is data on the expression of these genes in the same HS rats in nucleus accumbens, data that Hao Chen generated for all of those genes. I sorted it from high expression on, I believe it's log 2 of FPKM. So you can see the highest expression is that baculovirus IAP repeat containing 6 at a mean expression of about 10 log 2 units, which is very high. Well, it's high. It's not very high. Just to give you a sense, a mitochondrial gene or ribosomal gene or something like mallet would have an expression of about 14 or 15 on the scale. It's a gene. I looked at Birch 6. I'm not excited about it. It is primarily a developmental gene, but here again, you can see I'm biased already. I'm looking for something that's neuroscience-y, and I'm throwing it out because it's got a lot of developmental biology and a lot of cancer biology. So that's the risk that you have to take. So you, at this point, again, bias is rearing its ugly head. TTC27 was the first gene I looked at. I don't really know why. It's not inherently an interesting sounding gene to me. I knew nothing about it. The solute transporter seems very interesting. The other gene, the latent transforming growth factor beta binding protein 1, okay, seems interesting. But the first one that I clicked on happened to be that TTC27. And so let's see what happens. I'm going to skip this in the interest of time, leaving you guys some questions. This just shows some cool things you can do in GeneNetwork. I did an all-by-all correlation of all those transcripts. So you see a correlation matrix here of one gene or one transcript to another transcript. And I'm just looking at the correlation between 2 of those transcripts that happen to be located, or their cognate genes happen to be located in our QTL interval. And then I've done a fun thing. I've done a colored scatter plot in GeneNetwork with cofactors. So it's a 4-dimensional plot with the sex of the cases in the conventional color coding, and then the size of the dot telling you which batch it was. I think Hao had 3 or 4 or 5 batches. And you can see that there's really not a sex difference or batch difference. It's very prominent here, but that's mostly just for fun. But here's what happens if you go to GeneNetwork. And I almost fell over when I did this, but then I remembered what paper I'm dealing with. So in GeneNetwork, there's a tool called the Gene Wiki, which allows any user, you don't even have to be logged in. So if you want to put a Viagra ad into GeneNetwork, you are welcome to do so. You just have to enter your email address. In this case, there were no Gene Wiki entries for TTC27, but there was a gene reference into function that we harvest every night from NCBI. And it said clinical trial of gene disease association and gene environmental association from the huge navigator Homo sapiens PubMed. That's all that was in there. And I clicked on that PubMed link and I got what you see right there. Personalized smoking cessation interactions between nicotine dose dependence and quit success genotype score. So at that point, you could pat yourself on the back and say, most excellent. I've cloned the gene for Hao Chen and team, but there are at least half a dozen gotchas here. Those of you who know this work by George Ewell know that he tagged about, I think about 2,000 to 3,000 genes were tagged in this study. So I rummaged around in the study itself this morning and made an Excel spreadsheet, and I can't actually find TTC27 anywhere in his main list of about 300 genes that were associated with personalized smoking. So I think it's going to require a little bit more rummaging. But ideally, this would have been a very meticulous, low false discovery sort of study, and we would have grounds to take this gene more seriously as a candidate. Here's the Excel spreadsheet I generated today, showing you that in this case, he's got 338 hits. It was an interesting exploration of failure of links, by the way, to find these data. Here's the other, one of the other genes. So this is the RAS GRIP3 gene. Also pretty compelling. Here an interaction between dendritic spine plasticity, primarily in the hippocampus. So it's certainly relevant biology. But again, you can see that we're getting into the zone of narrative potential rather than real hardcore biology. So let's look at some minimally biased resources. So on each GeneNetwork page, there are resources that I regard as minimally biased: Gene Mania, Panther, String, BioGPS, GTEx Portal, Phenogen, Rat Genome Base, EBI, GWAS. These are not, they will have some bias, but they're not terrible. So here are the link to the Gene Mania page. So I've just gone, in this case, TTC 27 again. I've clicked on the Gene Mania link, and I'm looking at the network of TTC 27 in humans, or in, I'm sorry, that was rat, or in mouse, or in human. And Gene Mania allows you to look for known connections from protein-protein interaction networks, co-expression, other things. Very cool resource and a way to winnow down or evaluate candidates quite effectively. But any of those resource links there will provide useful data. I think the other one that's kind of interesting is the EBI GWAS catalog, in this case, again, for TTC 27. And if I were doing a hair loss QTL study in mice, I might be very happy because there's alopecia with an incredible p-value of 2 × 10⁻20, and it's apparently been replicated a few times. But of interest, there is a significant hit there next to the bottom: risky sexual behavior measurement, number of sexual partners. So I don't, you know, sex and nicotine, what's the difference? I don't know. Presumably some of you do know the difference, but I think that's a slightly relevant human GWAS hit that might provide some insight. Hippocampal atrophy at the very top, and intelligence, not very good p-values, but this provides one way to see whether you're barking up the totally wrong tree or not. And then phenome-wide association. So this is asking, what are, in human data sets, what are the known associations between TTC 27 sequence variants, SNPs, and lots of different phenotypes. And here you can see that there are 4 of these that are pretty damn good. So 10⁻⁵ and lower. And allergy, mycosis, optic atrophy, neither of those 3, or those 3 are not very interesting. Delirium, mentia, amnestic, and other cognitive disorders is not great, but is tolerable. GTEx Portal. So this shows us the expression for TTC 27 in normal humans, and I've highlighted the nucleus accumbens here. A total of 246 individuals were studied. And so you can easily go to GTEx and you can ask, what does TTC 27 co-vary with? And if it co-varied beautifully with DRD2, or some gene that you know is part of a molecular network or cellular network involved in addiction, you might be pleased by this as a candidate gene. All of the GTEx data is in GeneNetwork. So you can either study it in at the Broad, or you can study it at UTHSC and GeneNetwork. We've made it a little bit easier to do the correlation analyses and assemble your own collections. But I have to say that GTEx Portal, it looks cool. So nice little violin plots here. So summary. Yeah, I have a couple of summaries here, but the first one is I just want to encourage people that if you do try this and you treat the presentation as a primer, do send me bug reports. There's nothing more that I like than a grouchy review. If I get people that pat me on the back and say, great, 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 that's why we have errors like MM9 references 10 years later. So please send me bug reports. The more critical and nasty you are, I guarantee you, the more I will appreciate your input. If you have data sets that you would like to enter, we're delighted to do it. And coming again back to Joe's question, I will drive you crazy asking for metadata, because I have learned, and apparently Joe has too, that it's easy to get data from people. It's a major motion picture to get them to cooperate and give you good metadata. Just even abbreviations for their traits, a full description of their trait, because they're so interested in getting their publication out. They don't really want to dork around in your database system to get things solid for findable, accessible, interoperable, and reproducible research or reusable research. But that's my goal, is to really make all of the data that's in GeneNetwork reasonably fair, compliant. The methods I showed you of candidate gene evaluation, they're frankly seat of the pants, very ad hoc. They're very dynamic because the resources keep changing on a monthly basis. What we really need to do is hope that we get solid general AI systems in the next 10 years that can really help us navigate these data sets and rank order things in reasonable ways, but with tunable parameters, so that we humans are not left out of all of the fun. So you can imagine focusing more on CISI QTLs, or you're interested only in protein coding variants. So you want to be able to tune the parameters so that you're really only looking at high impact amino acid substitutions. And I just want to emphasize that it's always tempting to think that we are doing well and we sort of kind of deserve this word post-genomics to refer to our stage of life. But oh my God, we are so ignorant. We are not even out of diapers in terms of what we know and don't know about biological processes and their complexity. And I think for me, the giveaway is that we still use the horrible metaphor of mechanism. There is no mechanism. Everything in biology is a very contingent, evolutionarily fine-tuned mess, and we just have to get rid of the notion of necessary and sufficient and mechanism. Not going to win you any papers in neuron or cell anytime soon, but it's a metaphor that harkens back to steam engines and looms in Manchester. So,
Rob, there was a question.
Okay, I think I'm done here. Let me just say that, yeah, I'm done. There are a bunch more slides, and go through those if you want. Some test questions, if you want to see whether you can do this yourself or get your colleagues to do it, and then some worked examples. So that's it. So I'm done, and I will stop sharing my screen now. Oh, I do want to thank the heroes that have been coding on GeneNetwork now for 20 years. Right now, it's Zach and Piotr are the main heroes in terms of GitHub commits. We have a long way to go, but it's definitely worth forking. So if you think you can use an instance of GeneNetwork, you rebadge it, just use it. Okay, so that's the end, and I'm going to stop my share. Okay.
There was a question from Joe Nadeau that, are there any formal methods to identify the boundaries of the candidate interval?
There are formal methods, but they're not particularly accurate. And Joe knows this as well as I do. The old advice was take a 2-law drop. And then I think Carl Broman and others revisited that for most conventional F2 and back crosses and decided that a 1.5 log support interval was reasonable and captured the correct modeled locus 95% of the time. What I do is more, I think, reasonable but ad hoc. If I know the distribution of variance in a particular cross, which we don't, we do for the HS, we do in very gruesome detail for the BXD mouse population, then you can convolve the QTL support interval with the known position of, or the SNP density track. And so if you have a QTL that's 5 megabases, but half of it is a gene desert, you are fully justified in de-emphasizing the gene desert side and focusing on the gene rich, the SNP desert side. It's not formally sensible. If you see a great candidate over there, you'll want to study it, but it becomes a game of priors, essentially. And I'm sure you've been through this, Joe, as much as I have.
Well, we are about 3 minutes past our ending time, so maybe we can attack some of the other questions in that Q&A section on the website. So we'll have Rob formally respond to some of the other questions. So just a quick reminder that within the next 10 minutes or so, you should be getting an email from me with just a 3-question survey about the webinar and what you'd like to see in the future. And then within the next week or so, you should see an announcement about the next webinar. So what we're doing is the 2nd and 4th Friday of the month. So our next webinar will be June 12th. So thank you all for tuning in, and please use the links that are sent soon for both the survey and for all the material that we've covered today. Thank you very much, Rob.
Thanks. It's great.
Rob.
Yep.
Take care.